Dear students, I will be beginning with the second part of the apparatus, the equipments and instruments that are required for the microbiology laboratory. In the previous discussion, I had said about the glass apparatus, the metal apparatus and various instruments, the small instrument equipments that are used for microbiology practicals. Now here I will be discussing about the four main instruments that are used in the microbiology laboratory to carry out the various work. Whether it is in the college or in the industrial level, these are the four main instruments. The first one I say is the hot air oven. Now as the name itself suggests, it requires the hot air to heat up all the substances that are present within it. Now it is a process of sterilization. What is sterilization? Sterilization means to keep all equipments, all apparatus away from contamination by killing the microorganisms, especially the bacteria and the fungi that may grow. And it is very much required because in the practical process, whatever study we do, it should be contamination free. Here, the hot air sterilization or the dry heat sterilization usually do not require any moisture. It is moisture free. So we should keep in mind whatever substance we give for sterilization over here, it should be dry and not wet. So what is the process? As the washing process is done, that is cleaning, washing with soap, then we wash with the running tap water, then we rinse with distilled water. After that, we keep it, air dry it. After the air dry, wherever there is an opening, for example in the test tubes, in case of the pipettes, they are plugged with the cottons and then they are kept for sterilization in the dry heat in the hot air oven. How they are kept? Either they are wrapped with paper like the brown papers, or they are wrapped with the aluminium foil and are stored and kept into the various racks. Here if you see the racks, I have opened it and you can see the racks in the hot air oven. Now the sterilization can be done for the glasswares, the metal apparatus, powders. Remember that especially any plastic substances, the rubber corks cannot be sterilized by this method. What is the working principle? Means on what basis the dry heat will kill the microorganisms. It is the three methods of dry heat are the convection, the conduction and the radiation. I repeat, the convection, conduction and radiation. Conduction, if I say, the direct contact. If the temperature is rising over here inside the chamber, due to direct contact of the racks, the direct contact of the metal present over there, there will be a rise in temperature of the apparatus also. In case of radiation, the waves, the microwave oven, as you see in case of our microwave oven in the house also, household microwave oven, the wave, they causes heating of the substance. The most important over here, the convection process. In this convection, the air inside the entire hot air oven, they get heated up and this air circulated will cause the killing of the microorganism by three ways. One is by oxidation, second is by protein denaturation, and third is there is an increased amount of electrolytes over there. The concentration of electrolyte increases over there because no water is there, and that will lead to the death of the microorganism. And this is one of the best technique to remove the endotoxins of the microorganisms that are present. Here there are three different temperature and time conditions for heating or sterilizing in the hot air oven. If you want to sterilize for 30 minutes, the temperature in the hot air oven should be 170 degrees Celsius. Here on top there is a thermometer. Here just on the top there is a thermometer attached over there that indicates the temperature. If for 60 minutes sterilization is done, then the temperature should be at 160 degrees Celsius, and 150 degrees Celsius is required for 150 minutes of sterilization. So, immediate sterilization of glass material, suppose we have not sterilized in before, so we can use this type of dry heat sterilization of hot air oven. The next type of sterilization is the moist heat sterilization, and it is done by the autoclave. The autoclave is also called the steam sterilizer and acts on the principle of a pressure cooker at a home. Now this process was first discovered or invented by Charles Chamberlain in the year 1879. This concept of sterilization was introduced. Now normally the glasswares, the various media, then for example the various buffers, the sodium chloride, isotonic solution, that is normal saline, all these are sterilized in the moist heat sterilization. See, if you see over here, this is the entire unit of autoclave. If we open it from top, inside there is the space where we keep the water, and above the water we will keep all the substances that need to be sterilized, keeping in mind the water from outside does not enter inside the media or other substances. Now in this method, the concept that is used is the pressure. Now pressure is directly proportional to temperature. With an increase in the pressure, the temperature will also rise. Here the boiling temperature of water is 100 degrees Celsius, and we cannot increase it by any other means. We can only increase it when we will increase the pressure of that water. If the pressure of boiling water is increased, then only the temperature will also rise. So keeping this in mind, the entire steam sterilization is done. Here also, whatever we sterilize it, we first wash it, rinse it, and then wrap with the paper, and then we paper or aluminium foil, and then keep it into the sterilizer. And there are 3 stages of sterilization. The 3 stages of sterilization is the purge phase, and in this purge phase what happens? The steam displaces the air. Inside this entire body of the autoclave, there is air. That air should be displaced by the steam. What we do is, first we put all the items over there, we close the autoclave properly, then we switch it on and allow the steam to form. And there is a valve from where the steam comes out. Keeping to notice that the airflow should be at least a foot from there, so that a straight foot. That means the steam or the entire pressure is similar for that unit. The next stage is the sterilization phase, where we close the valve. When we close the valve, then what happens? The steam inside has a proper pressure, and this pressure increases the temperature. This pressure increases the temperature. Now this temperature, when it increases, that will lead to a proper uniform sterilization of the items. Once the sterilization is done for 20 minutes, then there is an exhaust phase where the slow release of the pressure takes place. And it is slowly, when the pressure is released, ultimate the temperature drops. Then we open the autoclave, we take out all the samples, we keep it in the air, or we keep it, do not open the paper or the aluminium foil, just keep it in the air or in the dry hot air oven to dry those substances. And whenever we use it, at that time you can open the air. Now here are the various sterilization cycle. You see the pressure over here, along with the pressure how the temperature rises, and accordingly we can sterilize it. The next one is the BOD incubator. Now the term BOD means biochemical oxygen demand or biological oxygen demand incubator, And this biochemical oxygen demand or biological oxygen demand incubator, what is this? That is the amount of oxygen that is utilized or depleted by a microorganism or a bacterium. And this is also called a refrigerated incubator or a low temperature incubator. Why? Because normally the incubator, which is not a BOD, the temperature over there is 37 degrees Celsius. That is the optimum temperature for the growth of most of the bacteria or fungi. Here what happens? Here we use the temperature range from 5 degrees Celsius to 60 degrees Celsius. That is both at the cooling temperature as well as a high temperature can be used as an incubator, and incubation of the microorganisms can be done, especially the bacteria. Here the cultures as well as fungi, here the cultures are stored, the temperature of the constant temperature for tissue culture that is required, that type of culture can be grown. Then the high temperature incubation can be done, low temperature incubation can be done, and it totally depends upon what is the oxygen demand of that bacteria. Suppose there is a change in the color of the media or the change in the color of the colony, that means the desired or the required oxygen species of bacteria has transformed into a separate species. And remember, normally oxygen is required for the aerobic bacteria, not the anaerobic bacteria. The last most important instrument of the microbiology lab is the laminar airflow, and this is also called the contamination free workstation or the sterile cabinet. This is actually the model, and here in this model we have a filter over here, a blower, the two light system, one is of UV light system, the other is the normal light system, a glass cover over here, and there is a Bunsen burner. The concept is, whenever you work over here, the first thing you should do is you switch on the UV lamp, keep it for 5 minutes, and please do not stay in front of the UV lamp. You switch off it and then you start your work. Again, after the end of the work, you are supposed to switch on the UV lamp so that if any microorganism is present in presence of the UV lamp, it will die. Then you should always start the blower for 10 minutes before you start an experiment, so that all the air that is present, particulate air that is present over there, it gets cleaned. Now the laminar airflow, this sterile unit, it is of two types. One is the vertical hood, the other is the horizontal hood. Now vertical hood and horizontal hood, what is the difference? The difference is the way the air is getting circulated into the chamber. If I see this one, see over here, this is the horizontal hood. What happens over here? The air from outside environment that is in the room is entering into the blower and it is coming over here. Then the air is passing through the filter. There is only one way of passing. It passes through the filter. The green arrows show that it is the filtered air. After it crosses the laminar hood where the workstation is there, it becomes red. That means it got polluted with the microorganisms or other particles that is present. Again this air comes out, or the environmental air comes and enters into the hood, and there will be a flow. So this is the horizontal type. See the air flow over here, it is coming from this way in a horizontal direction. And remember, not at all times it is coming from upside, it also comes from the back. The entry is also from the back where it directly enters into the workstation. Here if I show you the vertical hood, what is the difference in vertical hood? That is, the air directly comes in a vertical way. From the top it is entering. The air, the white arrows show the air from the room is entering. It is passing the green filter over here. The green air means the filtered air is going. Work you will be carrying out, and when the air is passing over here, it is getting contaminated with many substances, then it flows out. Again the circulation leads to the entry of the air from top, and this is the difference between a horizontal and a vertical hood. Normally The air inside the laminar flow should always be a class 100 air. That means, what do you mean by class 100 air? It is usually indicated that a vertical laminar hood has a class 100 and a horizontal has class 10,000 air. That means after filtering the air, there will be 100 particulate matter, and after filtering the air there will be 10,000 particulate matter, and class 100 air is much better than the class 10,000 air. So all work that is done in the microbiology laboratory, that is for inoculation, then subculturing, plating, all this type of small works that are carried out in this workstation, because it is the sterile unit. The entire room cannot be so much sterile like the laminar airflow. And remember, the entire air circulation is unidirectional. Whenever you use a laminar airflow, remember that keep it clean. After the work and before the work, please clean with 70% of isopropyl alcohol. After the alcohol dries up, then only start the Bunsen burner or on the Bunsen burner. Otherwise, the alcohol being inflammable, it may catch fire. So it is very much necessary we take the precautions and the safety measures, and to check that contamination does not go on either way when we are working on the microbiology lab. I have given you the references over here through which you can go, you can see the working principle properly, you can see the diagrams. Thank you, students.